Hello everyone. So this video is going to go over DNA structure as well as DNA replication and then how DNA gets organized based on what the cell is currently doing. So it's really important that you know what the letters DNA stand for. They stand for deoxyribonucleic acid. So deoxyribo gives you the D, nucleic gives you the N, and acid gives you the A. And actually, even though we write it as deoxyribonucleic, it's actually two separate words. Okay, so DNA, or deoxyribonucleic acid, is the material that carries your genetic information. And it's found in all living things, from the smallest bacteria to the weirdest bug you can imagine, to a blue whale, which is giant, and us, and everything in between. We all have some form of genetic material. The majority of us have DNA, and then there are a few weird offshoots that have a cousin of DNA, if you will, called RNA. So there are a lot of scientists that are credited with us knowing what we know about DNA and about nucleic acids in general, but the two that tend to be recognized the most often are James Watson and Francis Crick, because in 1953 they built this model, which you can see on the image, that kind of detailed exactly how DNA comes together and the shape that it makes. So along with Watson and Crick, before Watson and Crick actually, there was Griffith. Griffith's experiment kind of formed the basis for a lot of the future research that followed over DNA. So in his research, he knew that a substance derived from pneumonia, a bacterial that could kill things, could transform non-infected living bacteria with pneumonia to become an infected kind of bacteria. So he figured out, these bacteria over here have something that make them infected with pneumonia. These bacteria over here do not. But if I get them close enough to each other, it seems like whatever material is in the infected bacteria can also find a way to infect the non-infected bacteria. So it made him wonder, what is this material? How is it working? So he pretty much just studied how non-virulent or non-infected forms of bacteria could infect, or could become infected, by infected forms of bacteria. So the way that he did this, he worked with mice and he worked with these bacteria, and he infected the mice, live mice, with live infective bacterial cells. And that became the control. And because he knew if the cells are infected, they would find a way to infect the mouse itself, and the mouse would more than likely die. And that's exactly what happened. The mice died because of these infected cells getting in their blood. Okay, but he expected that. So then he took another live mouse and he infected it with live bacterial cells that were non-infective. And again, he expected that, hey, I'm not giving you anything that's deadly, so you should live. That's exactly what happened. The mice lived because there were no infected cells in their blood. So then he took mice and he injected them with cells that were infected, but that had been killed using heat. So the result was those mice lived because no live infected cells were in their blood, and that shows that the live cells were necessary to be virulent. So then he injected mice with heat-killed cells and live virulent cells, and the result was that the mice died because, again, they had those live virulent cells that had the infection in their blood. So he went through a series of these steps and figured out that there's some kind of material that could transform or cause death in an organism that was previously healthy. So after Griffith comes a trio, Avery, McLeod, and McCarthy. These guys did most of their DNA work in the 1940s, and they were the first ones to identify that this material was actually DNA. So through their work, they showed that DNA, and only DNA, was the molecule that was responsible for transforming these pneumonia bacteria from infected or from non-infected into infected. So before that, people had thought, well, maybe it's a protein, maybe it's RNA, because we already knew what those things were. But DNA was kind of obscure. We weren't really sure of the role that DNA played in living things up until that point. Okay, another group of scientists that continued to further what we knew about DNA were Hershey and Chase. And they did their experiment in 1952. And what they did was they worked with a type of virus that infects bacterial cells. And those viruses are called phages. Normally they're called bacteriophages. So we knew that viruses were not alive, but we also knew that viruses had the ability to make living cells sick. So Hershey and Chase worked with phages and bacterial cells and showed over time that the phages contained DNA. And it was the phage DNA that entered the bacterial cells and then infected the bacterial cell, allowing that cell to now produce more viruses on behalf of that virus, if you will. So they used the bacteriophage that was labeled with a radioisotope so that they could see it, they could find it or track it. And then they used a virus to infect the bacteria E. coli. And they could literally follow the path that viral DNA took inside of the living cell. So between Griffith and Avery and Hershey and Chase and all the people I've already mentioned, we established that DNA is the hereditary molecule of life. DNA carries the instructions. Okay, so now that we've had our history lesson, let's now start talking about the science of nucleic acids. We touched on this briefly when we talked about biomolecules because a nucleic acid is one of the 4 major groups of biomolecules. So remember, your nucleic acids are made up of the monomer nucleotides. And they are built by stringing or constructing chains of nucleotides together. Now remember, each nucleotide, especially in the case of, each nucleotide is going to be made up of 3 things: a sugar, a phosphate group, and a nitrogen base. In the case of DNA, the sugar is deoxyribose, the phosphate group is the usual phosphate group, and the base can be one of 4 things: an adenine base, a thymine base, a guanine base, or a cytosine base. Okay, so if you were to look at the image, this whole thing that I just put a box around, that is my nucleotide. So if I were to number these 1, 2, and 3, then here's my sugar, 1. This is my phosphate. I'm going to draw a circle around it. That's 2. And then my nitrogen base is right here. That is 3. Connecting the nucleotide to the one above it or the one below it. So I'm actually going to erase just a little bit of this circle that I've already drawn. And I'm going to switch colors here. So this bond right here that I'm now redrawn in green between the carbon and the oxygen, that is the bond that is holding that last nucleotide to the nucleotide above it. Oops. And this bond is a covalent bond known as a phosphodiester bond. Okay, and again, it's the bond between the carbon and the carbon of the sugar above and the oxygen of the phosphate below of the nucleotides that are next to each other, if you will. Okay, so that's one kind of bond that you are going to find in nucleic acids. And then in DNA specifically, because DNA is 2 strands and they're twisted together, they're held together in the middle by hydrogen bonds. But I'll show you that on a different picture. Okay, so I'm still going to push some history through where it needs to be applied. So I mentioned on the previous slide that there are 4 kinds of nitrogen bases: adenine, thymine, cytosine, and guanine. Because of a biologist known as Erwin Chargaff, we have what we call Chargaff's rules. So Chargaff did his experimentation in the 1950s, and he discovered that the nitrogen bases in DNA, the adenine, thymine, cytosine, and guanine, actually occur in specific ratios. Okay? And this is something that we still utilize and teach to this very day. Every high school kid knows Chargaff rules. They may not know that's what it's called, but they know what I'm about to tell you. So Chargaff discovered that 2 of the nitrogen bases are similar in size and in makeup, and then the other 2 are also similar to each other in size and makeup. And if you look at the picture, you can clearly see that. We have these 2 nitrogen bases that are double ring structures, about the same size, and then we have these 2 nitrogen bases that are single ring structures. So we call the double ring structures purine. We call the single ring structures pyrimidines. And the way I teach my high schoolers to remember purines is the letters A and G on the periodic table are the symbol for the element silver. So I make them write the words, I make them write the words pure, silver, and then we write purine, so pure for purines, equal AG, because AG is silver on the periodic table. So that's how I teach my high schoolers to remember this. So anyway, back to Chargaff. So Chargaff discovered that the number of purines were equal to the number of pyrimidines. However many A's I had the same amount of T's. How many C's I have the same amount of G's. So he came up with the rule that says that A equals T and C equals G. Really, A's and T's match together and G's and C's match together. They fit together almost like puzzle pieces. So if I have 10 A's, I must have 10 T's. If I have 20 G's, I must have 20 C's. Okay, so that's known as Chargaff rules, and it makes up what we call base pairing rules. A's go with T's, and T's go with A's, C's go with G's, and G's go with C's. Now, up until this point, we know all this information about DNA, but we are yet to discover what DNA looks like. Like Watson and Crick haven't actually done their work yet. Okay, so we're getting closer to that point. Now, Watson and Crick would be completely lost without a female scientist by the name of Rosalind Franklin. And I personally feel that she never gets any glory. And I guess because I'm a female scientist, I'm very attached to her in that sense. So I always make it a point to talk about her. So Watson and Crick had been working on this model of DNA, and it just wasn't fitting together. Something was stumping them. They could not make sense of it. And the problem is that they had the model flipped around backwards, so it wasn't fitting together. But Rosalind Franklin was using X-ray diffraction to study DNA at the time. So she was looking at this X-ray image of a DNA sample, and she realized that the sample, when you looked at it, made this X shape. And in the X shape, there were specific areas that were darker and lighter than each other. And what she realized is what she was looking at was a helical structure. So she was the first one to really realize that DNA had a helical structure. She made the discovery before Watson and Crick. And as a matter of fact, they used her imagery to finally make sense of their model that they had been building. They had the phosphates and the sugars on the inside of the model, and they had the bases pointing outwards of the model. And that's why it wasn't working, because it's the opposite way around. The phosphates and the sugars make up the backbone, the outside of the DNA model, and the bases actually meet in the middle. So again, here comes Watson and Crick, because notice they were working at the same time that Rosalind Franklin was working, so 1953. So They had got access to her research, not necessarily by ethical means, let's say, and they were able to finally discover what the structure of DNA was, that it's actually made up of these two nucleotide chains that are twisted together into a right twisted double helix. So as we know it, this is what DNA looks like. Two nucleotide chains that are twisted around each other, almost like a spiral staircase or a twisted ladder. And the chains are anti-parallel. They run in opposite polarity directions to each other. I'll explain more about what this means. But note what I'm saying. So here I have my phosphates and my sugars on the outside, here and here. And then I have my nitrogen bases making up the middle, okay? So when I talked about there being hydrogen bonds that hold DNA together, I'm going to use black here. These lines represent the hydrogen bonds that hold the nitrogen bases together. So if you look between C and G, you'd notice I drew 3 lines because there are 3 hydrogen bonds there. But between A and T, there are only 2 hydrogen bonds. So again, A and T go together, C and G go together. DNA is made up of these twists, and we actually know that a full twist, so from here to here, is made up of 10 base pairs, 10 sets of A's and T's and C's and G's. Okay, so now that we know more about DNA structure, let's talk about how your body makes DNA. Well, your body makes DNA through a process known as DNA replication. And why do we need to replicate DNA? Well, it's because your cells divide, right? Remember we talked about mitosis and meiosis, last week? So in order for me to have genetic information for the cell I'm about to make, I must first copy the genetic information that the cell has available. So why am I replicating? I'm replicating so that when cell division takes place, all of the cells will have DNA in them. When does replication happen? Remember, replication happens in the cell cycle, specifically in the S phase. Okay, so how does replication happen? Like, what does the process look like? Well, more DNA scientists. Mellison and Stahl in 1958 were the first ones to do work on exactly what does the DNA replication process look like. And they came up with 3 possible hypotheses. They thought, well, it could be a semi-conservative process, it could be a fully conservative process, or it could be a dispersive type of replication. Those were their 3 options. Okay, so here you can see on the picture what these 3 look like. So semi-conservative means I start off with my original DNA, and then the first time I make a copy, each DNA is going to be made up of an original strand and a brand new strand. Original strand and a brand new strand. When that goes through replication, then I'm going to have an original strand and a brand new strand on one of my copies, but the other copy is going to be fully made up of new DNA. That's what semi-conservative is. I'm keeping half of the current DNA, and I'm just making another half of it. So one half is old, one half is new. With conservative replication, I have my original DNA, and that DNA gets copied, and both of the new strands come together to make my second copy of DNA, and my original copy of DNA rejoins itself and goes back to being original. Okay? With dispersive replication, sections of my old DNA is incorporated with sections of my new DNA. And that's how both pieces are made up. So that was their theory. So now, of course, they had to test it. And they did so by labeling DNA and pretty much following to see where the labeled DNA went. And at the end of testing all of the three hypotheses, the experiment matched the semi-conservative hypothesis. So we now know that replication is a semi-conservative process. So what does this mean? Well, it means that the first thing that has to happen is my DNA molecule, excuse me, has to break the bond, the hydrogen bonds holding it together. Then it has to unwind into two separate strands. Each of those strands can then act as a template, and because the bases pair up in a certain way, G with C, C with G, A with T, T with A, then we could use this strand to figure out what bases should be filled in for the new strand. So because we are using this original strand as a guide or as a template, it means the new strand, which we're using red in this picture to show, is going to be complementary to the original strand. A with T, T with A, C with G, G with C. So you do that on both sides. Each strand of the original DNA would become a template for the new strand, the two new strands that we are building. So each double helix at the end of it will be made up of one parent strand or old strand, which we're showing in gray, and one new strand, which we are showing in red. Okay? So that's why it's semi-conservative. Half of the original material is still there, and half of the strand is also new material. Excuse me. Now, DNA has to be built in a very specific direction. So this is something that I need you to know. DNA is synthesized or built in the 5' to 3' direction. Now, we've already talked about primes a little bit, but it was way back in biomolecules, so I might do another diagram for you in a second. So when we are talking 5' and 3', you have to think back to what the sugar of a DNA molecule looks like. So it looks kind of like that. Everywhere those lines meet, there is a carbon molecule there. Okay? Now, DNA, in terms of building phosphodiester bonds, they only happen, technically, the 5th carbon is actually sitting above the 4th carbon. So this is carbon 1', 2', 3', 4', and 5'. So when it comes to building phosphodiester bonds, we only build them between the 5' carbon and the 3' carbon, okay? So on this next molecule of DNA, we would have to attach the oxygen of that phosphate group to the 3' carbon. We can only attach it to 5' or 3'. So DNA specifically is built from 5 to 3'. We always add on the 5' end. Okay, so now we're going to talk about all of the enzymes involved in DNA replication, and there are a lot of them. You are going to have to remember them. So I'm not going to go through this list in the order that it's written. I'm actually going to go through it in the order of how these enzymes function, okay? So the first enzyme we're going to talk about is helicase. Helicase is an enzyme that is going to open up the DNA and untwist the two strands from each other so that we are no longer a double helix, but we have two single stretched strands. And it does so by breaking the hydrogen bonds that hold the nitrogen bases together. Okay, so helicase is one of the first enzymes that will start its job. The problem is DNA is twisted up. So if I just start opening it up, I'm going to have areas that get super coiled up in front of helicase, and I may even have areas that get super coiled up behind helicase. So that could create an issue where my DNA could break or become tangled. To prevent that from happening, we utilize an enzyme called topoisomerase. Topoisomerase's job, is to move ahead of helicase and make cuts so that the DNA can unwind a little bit and then rejoin that DNA so that no information has been changed. So it moves what we call downstream of the helicase enzyme, or which is part of the replication fork, and it helps to relieve the tension that helicase causes by opening up DNA kind of from the middle. Now, once those strands of DNA open up, we want to make sure that they stay away from each other so that they don't just rejoin. What's going to help us with that are not enzymes, but proteins called single-stranded DNA binding proteins. So they attach themselves to the separated strands and they hold them open. They keep them apart. Excuse me. Oh, I'm so sorry. So now that DNA is opened up and we can access the information in the nitrogen bases, we can start building our new strands. Well, in order for us to do that, we need a little bit of a primer. So I need you to think of a real life primer, like if you've ever painted a wall or repainted a table. You normally put down a layer of primer first because it helps the paint that you're going to put down to stick and it gives better color payoff, right? Well, DNA has a similar vibe. So I can't build DNA into thin air. I have to have something to attach it to. So we actually start off with a little bit of an RNA primer, and there is an enzyme, it's not written in, so I need you to add this please, called primase, that is going to lay down a very small sequence of RNA, usually only about 5 nucleotides long. And that is going to provide us a free 3' end that we can attach DNA to. Because remember, DNA has to be built 5 to 3'. Okay, so now that I have my primer, I can actually start building my complementary strand. And it is the job of DNA polymerase 3 specifically to lay down the new nucleotides to the DNA strand. And then now there are 2 strands of DNA working at the same time or being built at the same time. But remember, the strands are anti-parallel to each other. So one strand runs 5' to 3', and the other strand runs 3' to 5'. Because one of the strands runs in the opposite direction, it means that its new template doesn't get laid down continuously. The complementary strand isn't going to be made or built continuously. It's going to be built in sections, and I'll talk more about that in a second. So those sections are called Okazaki fragments, but before we can finish replication, we have to make sure that each section has been joined to the section in front of it. So that way we get 2 fully complete complementary strands of DNA. So I say all of that to say there is an enzyme that we kind of call DNA glue. It's called DNA ligase. And its job is to link all of the newly synthesized DNA pieces together so that we have 2 continuous strands. Okay, so let's look at this in picture form. I'm going to switch to my laser. So here I have DNA helicase. DNA helicase is going to move in this direction. It spins as it moves, and as it does that, it breaks apart those hydrogen bonds, creating 2 straight strands of DNA. Excuse me. These little blue dots represent the single-stranded binding proteins. They're going to stand there and hold those strands open so that they don't retwist. Here is topoisomerase, which is moving ahead of helicase, and it's just making little cuts so that this DNA can unwind and then it'll rejoin. So that way it's not too twisted up here in this area where it will break. Now, in order for us to build DNA, we must first have a little bit of primer. So here is primase, and it's laying down just a little bit of RNA primer. And as long as we have a free 3' end, we can then build DNA. So on this side, this is always going to be the 3' end. As we open up, it's always going to be 3' here. So if we switch down to this part of the picture, once I have 3' available, here comes DNA polymerase 3, and it is going to move along the DNA. It is going to read off the bases, and add in the correct matching nitrogen base so that we have a complementary strand being made. Now on this side, it's a little bit more difficult because since this is running in opposite direction to the strand above it, I have to have enough of this open because I'm not actually going to build the DNA towards the right. I'm going to build the DNA towards the left because the 3' end is going to be over here. So I have to open it up a little bit, put down the primer, and then build DNA back this way. And then we wait until more DNA opens up, and then again we build backwards. So this strand is being built backwards, but this strand is being built continuously. There's no need to wait because the 3' end is over here. It's already available to us. Okay, so after this has happened for a little bit, here you can kind of see what I was talking about. So here, this complementary strand is built continuously, but this complementary strand is built in pieces. So every time I have a new primer, like here and here, I will have DNA that follows. But see, this DNA is not joined to this DNA. So that's where the work of ligase comes in, and ligase is going to come in and connect all of those DNA pieces. It'll also remove the RNA and replace it with, so that DNA can replace the RNA, because we can't have little sections of RNA in our DNA either. Okay, so again, you can see, built continuously. We've already had ligase come in. Ligase is connecting everybody. It'll have to come back and connect this section as well. So the strand that's built continuously is known as the leading strand, and the strand that's built in sections is known as the lagging strand. Each of those sections are known as an Okazaki fragment. They were named for the Japanese scientist who discovered them. Okay, so let's go through a quick summary of DNA replication. Remember, all of this is happening in the S phase of the cell cycle. So helicase is going to unwind the parent DNA. The single-stranded binding proteins are going to attach themselves to the unwound DNA, so to stabilize it. The leading strand is going to be made continuously in the 5' to 3' direction by DNA polymerase. The lagging strand is made discontinuously, so in pieces. Primase is going to lay down a short piece of RNA primer, which will be added on by DNA polymerase to form an Okazaki fragment. And after the RNA primer is replaced by DNA, DNA ligase is going to come back and join all of the Okazaki fragments into one continuous strand. So the most concerning part of this is always the continuous versus the discontinuous building of the two strands. So let's talk about that. It goes back to the fact that DNA is anti-parallel. Okay, so if this is my DNA strands, one strand has the 5' carbon on this end, and at the very end of the strand, the last carbon is going to end on a 3'. The opposite strand is going to start with the 3' being exposed on the top, and then the last carbon will have the 5' exposed on the bottom. This is what we mean by anti-parallel. Okay, so I run 5 to 3, or I run 3 to 5. They do run in the same direction. Okay, so as the DNA unwinds, one of your template strands is going to run in the 5 to 3 direction, which allows us to just build DNA without even thinking about it. But the other one has to be copied in short sections until the entire piece has been copied, and that's why it's discontinuous, okay, because it runs in the opposite direction. All right, so this image shows you how this top strand is going to be our leading strand, because look, here's my free 3' end. Once I have a 3' end, I can then attach DNA. So because this end is 3', it means it'll always be 3'. We just keep on attaching. But this one down here, even on this side, I have a 3'. So technically I have a 3' here, but I can't build backwards this way. Whoops, sorry. Because DNA is opening up in this direction, okay? So once I build, the 3' will be here, but in terms of what's open, it's just my 5' end that's available. So I have to open up enough that I can start off my primer. Here's my 3' end, and now I go this way. I wait for it to open up a little bit more. I lay down some more primer, I build. I lay down some more primer, I build. The issue is, because I have to wait for DNA to open up, it takes a little bit before enough space is made that I can build DNA on this side because of that 3' rule. There has to be a 3' end available for DNA to be synthesized. So once it's done at the end, all we do is we go back and we zip it all together using ligase. So again, if you look, here's my 5' end, here's my 3' end, but I couldn't build until I had enough of this exposed so that I could go in this direction. Okay, so here is an image showing you with the nucleotides pictured. So here's my parent strand. Notice 5' carbon, 3' carbon. Okay, on this 3' end, so if it's 3' on this side, it means it has to be 5' on this side. So once I lay my nucleotide down, this 3' is empty, so I can attach another nucleotide right here. Again, this 3' is empty, so here comes my new nucleotide. I'm just going to attach it right there. This phosphate is just going to get attached right here to this carbon, and that just keeps going. So to make replication a very efficient process, it actually happens from multiple points of origin or multiple starting points, especially with more complex organisms. The more DNA you have, the longer it's going to take for us to copy all of that DNA. So these sites that replication starts at are known as origins of replication, or ORI sites. And literally that's what they are. It's where the process of replication takes place. And they are bidirectional. So if you look at the image, they make up these replication forks right here and right here, and they move in 2 different directions, opposite from each other. So you can have multiple sites and they will just move until the DNA finds each other, and then we join it all together. But it makes the process very, very efficient and very quick. Okay, so let's do another quick little review. So DNA is always synthesized 5' to 3', but it's read 3' to 5'. Because of this, one of the strands is going to be a lagging strand, which is going to be synthesized discontinuously into these short fragments that we call Okazaki fragments. The leading strand, however, is synthesized continuously. DNA is anti-parallel, meaning one strand is going to run from 5' to 3', while the other strand is going to run from 3' to 5'. DNA synthesis or replication is bidirectional, based off of the original ORI sites. So it moves in 2 different directions, and this helps the process to be quick and efficient. So once I've copied my DNA, what next? Well, there's an interesting little addition that we find in eukaryotic genes or eukaryotic DNA called telomeres. So a telomere is an expandable, non-coding region of DNA, and it's found on the very ends of your DNA molecule. So they're non-coding because they contain repeated units of a specific 6-base sequence, and that sequence is TTAGGG. Okay, so that just gets repeated over and over again. Well, what is the point of a telomere? The telomere is there to protect the gene from being eroded. The process of replication cuts off the bottom part of the DNA a little bit every single time it happens. So the more replication takes place, the more that bottom end of DNA gets chopped off. So by adding a filler there, by adding a protective end, even though the end is still going to get chopped off, it's not informational. It's not important coded information. It's not genes. It's just gibberish pretty much. Okay, so if I have a telomere section, I have to have something build that telomere section, and the builder of telomeres are the enzymes called telomerase. So telomerase is the enzyme that is going to lengthen the telomeres. It was discovered by a female biologist named Elizabeth Blackburn, and she recognized that it's a short molecule of RNA that serves as a template for new telomere regions, and then it's just these repeated 6 bases over and over again. It's not present in most cells of higher organisms, and older individuals tend to have shorter DNA in somatic cells. So especially in things like us, our telomere regions are very short, if they exist at all, and then after a while we just stop making them and we start chopping off shorter DNA. We're making shorter DNA because we're chopping off some of those extra sequences. Okay, so I just copied all of the genetic information in my body, and it seems like a fairly quick process. What's preventing my system, my cells, from making mistakes? Well, truthfully, nothing is. Mistakes will be made, and it happens all the time during replication. Sometimes the wrong base gets matched up with, the wrong base on the complementary. That should have been strand. Sorry about that. So when that happens, that means that we have a mistake in the genetic information that could be disastrous. So it's going to have to be corrected somehow. Okay. So the good news is that DNA polymerase has a proofreading mechanism. DNA polymerase actually is made up of 3 different sections, if you will. Section 3 is the part that builds new DNA, but section 1 is the part that proofreads the newly built DNA. So DNA polymerase is going to run back down that newly built section, checking for any mismatches. And when a mismatch happens, when it finds a mismatch, it is going to go ahead and cut that mismatch out using an enzyme, and then it will replace the missing nucleotide with the correct one. So remember, A with T, T with A, C with G, G with C. So there are only about 1 in a million errors occur in your DNA sequence. In comparison, when we make RNA, we only have about 1 in every 1,000 errors, because there is no repair mechanism in RNA. Okay, so even though mistakes happen, it's still a fairly efficient system. So let's talk again about the mismatch repair. Okay, let's look at the picture first. So when there's a mismatch, it doesn't actually fit together, not flush. Think of a puzzle piece. If I put the wrong 2 pieces together, it's very obvious. There's a bump there that tells me, hey, these 2 pieces don't go together, right? So it's the same thing on the DNA. There's going to be this bulge that tells me, hey, there's a mismatch here. So that bulge is called a distortion, and it's created because of those bases that are in the incorrect position. So repair enzymes are going to go find the bulge, and they're going to cut outside of the bulge. So they're going to cut a little above the bulge and a little below the bulge. This ensures that the actual mistake is taken out. So they'll remove it, and then they'll fill in the gaps using DNA polymerase 3 with the proper matching bases, the way it should have been done originally. And then after that happens, there's still going to be a little space. So DNA ligase is going to come and seal the pieces together so that, again, we have nice connected DNA. Okay, so this is just an example of DNA repair. We see it a lot in skin cells due to exposure from UV radiation. So it's called a thymer dimer, and it creates this weird bulge or distortion right around here, and eventually it's either replaced or it's not, just depending on how often that section is proofread, but the old skin slashes off and it gets replaced with new skin. So once that has been corrected, then no longer have an issue. So everything we've talked about thus far has been mostly focused on eukaryotic DNA. With prokaryotic DNA, their chromosomes are actually circular and they're complete. They're closed. So one closed circular piece of DNA. And remember, with prokaryotes, there is no nucleus. Everything's just kind of floating around in the general center of the cell that we call the nucleoid region. So replication is actually going to start from a single point of origin. With eukaryotic replication, there's lots of places for it to start, but with prokaryotic replication, it starts from a single point, and then it still moves in two directions until all of the DNA has been copied. Okay. Now, in many bacteria, they have small sections of DNA called plasmids that are capable of replicating independently of the rest of the DNA in the cell. So sometimes replication is happening in the original cell's DNA, the main DNA, but replication can also be happening in these little small pieces of DNA that bacteria can pick up from their environment that we call plasmids. All right, that's it for this video. Talk to y'all soon.